Assalamualaikum and greetings. My name is Najwa Arifah binti Idrus and I will be presenting my final year project titled Biodiversity Unveiled, an in-depth study of tree diversity in Sijangkang's mangrove ecosystem. For background of study, mangrove forests are among the most productive ecosystems on Earth, with growth rate comparable to tropical rainforests. These ecosystems support a rich variety of aquatic and terrestrial species across various taxonomic groups. Furthermore, the Rhizophoraceae family, which include Rhizophora, Bruguera, Cerops, and Candelia, is known to dominate to many mangrove habitats and consists of some of the oldest flowering plant species. Despite existing research, mangrove ecosystems are still not fully understood, especially as ongoing environmental degradation continues to impact them. The diversity of tree species plays an important role in supporting the resilience and stability of the mangrove forest ecosystem. The aim of the study is to identify the different tree species present in the Sijangkang mangrove area. It also seeks to evaluate the species richness and evenness of the tree population within the site. Finally, the study aims to determine which tree species are dominant in the Sijangkang Recreational Mangrove. We move forward to problem statement in which the Sijangkang Recreational Mangrove is a valuable ecological area that faces increasing threats due to human activities such as illegal dumping and pollution. These actions have resulted in serious waste accumulation and environmental damage. Although some conservation efforts have been implemented, there remains a lack of comprehensive data on tree diversity in the area, particularly in terms of species richness and abundance. This gap highlights the need for improved biodiversity research to support effective protections and management strategies. This research was carried out using 5 steps. These include in identifying the study area, designing the sampling method, species identification, biodiversity analysis, and finally, data analysis using biodiversity indices. The study was conducted at Sijangkang Recreational Mangrove Forest, which is located in Selangor. This map from Google Maps shows the locations of the mangrove forest where data collection was done. Next, a total of 4 quadrats measuring 10 by 10 meter were set up for this study. These quadrats were used to sample and record tree diversity within the mangrove forest. The setup ensured that the data collected was systematic and consistent. Moving on, the identification and documentations of tree species were conducted within each quadrat. Species were identified based on visible features such as leaf and stem. To ensure accuracy, Google Lens were used and also supported by professional guidance. Moreover, after identification, all tree species were labeled and documented carefully to ensure the reliability of the data used in the biodiversity analysis. For data analysis, PAS software was used. This allowed me to calculate several biodiversity indices such as the Shannon-Wiener Index, Simpson Index, Pielou Evenness Index, and Margalef Richness Index to evaluate diversity more comprehensively. Now, I will present the result and discussion based on the data collected and analyzed from the study area. This section includes finding on tree species composition, biodiversity indices, and the dominant species identified in the Sijangkang mangrove forest. A total of 131 individual trees were recorded, consisting of 19 species from 12 different plant families. Quadrat 2 recorded the highest abundance with 63 trees, followed by Quadrat 1 with 28 individuals, Quadrat 4 with 21, and Quadrat 3 with 19. Although Quadrat 2 had the most trees, Quadrat 3 and 4 showed the highest species richness, each with 7 different species. Following that, the species distribution was also analyzed by plant family. 7 main plant families were identified. The Rhizophoraceae family was the most dominant, comprising 48 individuals of 37% of the total populations. This was followed by the Araceae family with 44 individuals, accounting for 34%. Combined, these two families represented more than 70% of the total number of trees recorded, indicating their strong presence and potential ecological influence in the mangrove area. Apart from species and families, the trees were also grouped according to their growth stages. The trees recorded were characterized into 3 growth stages: pool, sapling, and seedling. The pool stage had the highest number with 61 individuals, followed by 36 seedlings and 34 saplings. This indicates that the mangrove ecosystem is actively regenerating and supports natural forest development. To assess the biodiversity of each quadrat, several indices were calculated. Quadrat 3 recorded the highest diversity with a Shannon-Wiener Index of 1.825. It also showed the highest richness based on the Margalef Index, as well as the highest Evenness value of 0.886. On the other hand, Quadrat 1 had the lowest value for diversity and richness, suggesting limited species distribution. The Simpson Index further supports this finding, where Quadrat 3 had the lowest dominance value, indicating it was the most balanced and biodiverse among all quadrats. Next, we look more closely at which species were most abundant in the study area. The most abundant species identified in the study was Nypa fruticans, which had 44 individuals. This was followed by Rhizophora mucronata with 29 individuals and Scyphiphora hydrophyllaceae with 14 individuals. These species appeared to be dominant in shaping the mangrove vegetation structure. In conclusion, a total of 19 species from 12 families were recorded. Biodiversity indices revealed that the Quadrat 3 had the highest diversity and balance, while Quadrat 1 had the lowest. The most dominant species was Nypa frutican, followed by Rhizophora mucronata and Scyphiphora hydrophyllaceae. This reflects uneven distributions of species within the area. Lastly, for future research, it is recommended to expand the survey area and collect more detailed data to get a clearer picture of tree diversity. It is also suggested to place more quadrats in different parts of the mangrove, such as near the river, in the mid-intertidal zone, and close to the sea or inland areas. This approach would improve the accuracy of the results and help with better conservation planning. These are the references used in the study. All sources were chosen to support the methodology, analysis, and discussions of tree diversity in mangrove ecosystem. That is all from me. Thank you.